So let's start glycolysis. Uh, you know that glycolysis is the oxidation of glucose to pyruvate. First of all, glucose enters into the cell. Glucose enters into the cell via transporter, which is called glucose transporter or GLUT. There are four types of GLUT: GLUT 1, 2, 3, and 4. GLUT 1 and 3 are present in most tissues, brain, and red blood cells, and they mediate the basal uptake of glucose, while GLUT2 is present in liver and pancreas, while GLUT4 is present in muscles, skeletal muscles, and adipose tissue. So glucose enters into the cell, and the glucose is a naughty molecule. It doesn't remain in the cell. As soon as it gets into the cell, it leaks out of the cell. So we need to trap this glucose. So glucose is trapped by an enzyme which is called hexokinase, right? And it is converted into glucose 6-phosphate. Here, an important thing: whenever the word kinase comes, ATP is either converted into ADP or ADP is converted into ATP. So whenever there is phosphorylation or dephosphorylation of ATP into ADP, so the enzyme is kinase. Then glucose 6-phosphate makes fructose 6-phosphate by an isomerase. Fructose 6-phosphate makes fructose 1,6-bisphosphate that is cleaved to make glyceraldehyde and dihydroxyacetone. Glyceraldehyde 3-phosphate then makes 1,3-bisphosphoglycerate, and that makes 3-phosphoglycerate, and then 2-phosphoglycerate, and then 2-phosphoenol pyruvate, and finally the pyruvate. So this is the whole story. But you are not supposed to worry about all this pathway, and it's not relevant to learn all this pathway. So what is relevant is that you see all the reactions of glycolysis are reversible except the three reactions. And what are those three reactions? First one is this: the first step, when glucose is converted to glucose 1-phosphate. This is the first step that is irreversible. And the second step, when fructose 6-phosphate makes fructose 1,6-bisphosphate. This is the second step, which is irreversible. And the last step, when phosphoenol pyruvate is converted into pyruvate, that is the third irreversible step. And if we could reverse these three irreversible steps, we can get glucose from pyruvate. And this is what actually happens in gluconeogenesis. In gluconeogenesis, we get glucose from pyruvate. This is an important point. You have to remember that there are three irreversible steps in glycolysis, and if we could reverse those three steps, we can get glucose back from pyruvate. All right. Another important thing to remember is that, see that here ATP is converting into ATP, we get kinase, and here again ATP is broken down to ADP, and again the enzyme is kinase, which is phosphofructokinase, and this is the rate limiting step of glycolysis, PFK1. It's not JFK1, John F. Kennedy, it is PFK1. So PFK1 is the rate limiting step for the glycolysis, and this is indirectly activated by insulin. Because fructose 6-phosphate first makes fructose 2,6-bisphosphate, and then fructose 2,6-bisphosphate activates PFK1. So actually insulin is responsible for activating this step. And another important thing is that whenever the word dehydrogenase comes, it means NAD is going to be reduced to NADH, or FAD is going to be reduced to FADH. So there is dehydrogenase. So you see that here kinase, hexokinase, phosphofructokinase, and now again you see that here again ADP is converting into ATP. The enzyme is phosphoglycerate kinase, and in the last step pyruvate kinase, and again ADP is converted into ATP. Whenever the word kinase comes, it means ADP is converting into ATP, or ATP is converting into ADP. And whenever the dehydrogenase comes, this means it is going to remove hydrogen from the substrate. It is going to remove hydrogen from the substrate, and those hydrogens are going to NAD or FAD to make NADH and FADH, right? So this is an important step. As I told earlier, that the net result of glycolysis is that we get 2 NADH and 2 ATP. It's a little tricky, but really very easy. You have to just know that glucose has 6 carbons. And from 6 carbon glucose we get 6 carbon fructose. But once the 6 carbon fructose is broken down to glyceraldehyde and dihydroxyacetone phosphate, we get 3 carbon glyceraldehyde and 3 carbon trihydroxyacetone phosphate. You remember from my summary that these are the trioses. And this dihydroxyacetone phosphate again makes glyceraldehyde. So in effect, we are getting two glyceraldehydes from one glucose. And once these two glyceraldehydes are processed in glycolysis, we get 2 NADH. And we get 2 ATP here. And we get 2 ATP here. So actually there are 4 ATP. But in the initial process, these 2 ATPs already used. Here number one at one place, you see that ATP is used in here and the second ATP is used in here. So in effect we get 4 minus 2, that is equal to 2 ATPs, right? And we get 2 NADH. And now another important point is that this NADH which is being produced here, these two NADH which are being produced here, they have to be oxidized back to NAD, and this happens in mitochondria during electron transport chain in the presence of oxygen. But if there is no mitochondria or there is no oxygen, for example, there will be no oxygen in case of hypoxia or anaerobic respiration, and there will be no mitochondria in case of red blood cells. But in that condition, again, we have to oxidize these NADH back to NAD. And this happens when, and this is anaerobic. In anaerobic conditions, number one, you have to remember two things. There is no oxygen or there is no mitochondria. There is no mitochondria in case of red blood cells, and there is no oxygen in case of sternus exercise or anaerobic conditions. In that condition, NADH is oxidized back to NAD. NADH is oxidized back to NAD by the conversion of pyruvate to lactate. That is the reason pyruvate is converted into lactate, to oxidize this NADH back to NAD in the absence of mitochondria or oxygen. And this lactate, as I explained in my first video, this lactate goes to the liver where it is converted back to pyruvate in Cori cycle, right? So in summary, the pyruvate we got from glycolysis. In addition, we got 2 NADH and 2 ATP. And here, this pyruvate enters into the mitochondria, and pyruvate is converted into acetyl-CoA. The reaction is called link reaction, and pyruvate dehydrogenase. Again, the enzyme is dehydrogenase, so it means NAD has to be reduced to NADH. We see in a while. And then this acetyl-CoA goes to Krebs cycle, in the TCA cycle, or it can be used for fatty acid synthesis. We'll see in a while. Alright, and the last thing to point in the glycolysis is that dihydroxyacetone phosphate that is produced during glycolysis, this is important to produce glycerol 3-phosphate. Again, the enzyme is dehydrogenase, so NAD has to be reduced to NADH. And this glycerol 3-phosphate is important in triglyceride synthesis and fat synthesis. Glycerol plus fatty acids make fat or triglyceride. And this glycerol 3-phosphate is also important for electron shuttle. Why this electron shuttle? This electron shuttle is important because these NADH are produced in cytoplasm. So the electrons of NADH cannot go directly to the mitochondria. So they need a shuttle, and that shuttle is provided by glycerol 3-phosphate. So these are the important things in glycolysis. Again, I revise, there are 3 irreversible steps that need to be reversed to make glucose again from pyruvate, and that makes gluconeogenesis. Another important point, the pyruvate under anaerobic conditions makes lactate, and whenever the enzyme kinase comes, ATP is going to be phosphorylated to ADP is going to be phosphorylated to ATP, or ATP to ADP. And whenever the enzyme dehydrogenase comes, it means NAD is going to be reduced to NADH and FAD to FADH2.